Okay, so with that inspiration from an Olympic gold medalist and a record holder, we're gonna keep that spirit of competition going with a live debate in which all of you are going to choose sides, as I mentioned, okay, in my opening remarks. So without further ado, I'd like to ask Suzanne Pavon, Rashad Massoud, and Sana Syed to please open up the debate. Tell us the rules and take it away.
Thank you, Tim. It's really a privilege and honor to be here today. And what an inspiration session we just had. I could not stop myself from taking so many quotes, but definitely this is going to stick that you need to open up mentally. So physically, things will follow. So thank you for this. Today, we are going to have a very, very interesting topic and a debate that's close to my heart and field. I am Suzanne Pavon. My background, I'm a PharmD, and I did my doctorate in oncology, started my career in hospitals, and worked in pharma, biotechnology, and CRO like Novartis and Piramel and Sanofi Genzyme and TFS. And I'm also on a board of director of Iethico. And today, my role is to be the chairperson for our debate, clinical.
Honey,
What is the gold standard for our clinical research? So, our controlled clinical trials, the gold standard for research and learning. And we will have someone who's going to be defending the motion, and that's my colleague Sana Syed, who's going to introduce herself and defend her position after this introduction. And following, it's going to be my colleague Rashad Massoud, who's going to be opposing this motion and also introducing himself. After this, we're going to have 10 minutes in which we will open the floor. And we would all love to hear what's your comments, what's your opinion and thoughts, or even questions. Are you with the motion or against the motion? And then we will open to summarize for both Rashad and Sana. And after the summary, please, I would ask you to cast your votes. We were going to use the WOVA app, and then we will announce the winner. So without further ado, Sana, the floor is yours.
Thank you, Suzanne. So should I share my screen to take this away? I just want to make sure. All right. So give me 1 second. So while I'm opening up the presentation itself, just to briefly summarize, as Suzanne had mentioned, that my name is Sana Syed. I am a neurologist in Boston, and here we go. I think you can see my screen. So I am a neurologist in Boston, and I work in the drug development research space. So a little bit different from medical affairs, but we do work closely with the medical affairs colleagues. I am involved more in the design and running of clinical trials. I work for a pharmaceutical company as a global clinical lead, and I have primarily focused on demyelinating disorders in my career, but I have worked in other disease areas like neurodegenerative and psychiatric disorders as well. I have mainly lived in the space of trial design, new drug approvals, and I have worked a little bit also in the digital space as well for endpoint development. And today, my discussion is centered on showing how randomized controlled trials are indeed the gold standard for research and learning. And I am obviously supporting this motion. So I will continue the conversation by giving you a brief overview of what randomized controlled trials are, like the scientific methodology behind it. I know a lot of you are probably familiar with this. However, I thought like a brief intro would help. This schematic is basically intended to show how a very standardized design works. Okay, some of you may already know that there are different ways of approaching clinical trials, and there are complexities in the design as well. But the main idea that I want to relay from this schematic here is that you have a group of patients and then you randomize them in 2 treatment groups. This is extremely important because the randomized piece is the core statistical principle that takes care of any biases and whatnot, and allows the scientific question to be answered appropriately. Now, the 2 treatment groups are blinded, but one of them's getting the drug under investigation, and the other one is a comparator arm, whether it is a placebo or another drug that is approved for the same condition. And then we measure the efficacy. So we have preset endpoints, and we check how the patients did in the treatment arm and the control arm. And I think for this audience, you know that even the control arm, which may even be a placebo, sometimes there's a placebo effect as well. So we have to have a certain amount of people that offset that possibility as well. So this is just like a very basic design framework. Now, I want to also highlight, like, sure, I showed you a classical trial design. It's randomized, double-blind, placebo, or, active controlled. A bunch of endpoints are needed. You need a certain critical sample size. But what happens when you are faced with different types of diseases? So with every illness, it comes with its own set of complexities. And I'm using rare disease here as an example. Now, what do you get when you're in the rare disease realm? You have really small patient populations, and I think Suzanne has experience in this that she'll highlight for us. There's usually a huge unmet need because the rare disease, the less trials you have in them a lot of times, unfortunately, and the diseases tend to be more complex as well. So the trial comes in, the clinical trial comes in, and it's supposed to address these nuances. It has to optimize the sample size to answer the question, but at the same time be able to recruit the critical number of people that you need to answer that question. And when there's a high unmet need, you don't want people on that placebo arm if there is no other active control. You want to make sure that they're all getting the opportunity to be at least exposed to a potential treatment. When the need is high, you have to optimize these types of things. So you go into a crossover design, you change the randomization ratio, what have you. And then if the disease is complex enough, then you wonder if there's even endpoints available that you can use to measure what your scientific question is, basically. So you have to think about all of these things when you're dealing with the trial design. So, and depending on the disease area. Now, why do I bring all of this up? Because the idea is that what we're trying to do with a clinical trial is we're trying to translate the biology of the drug to the patient population, so the real world patient experience. Now, the other factors that are there are like the real world factors. And sure, those are also important. They have an impact. But the mainstay of a trial is translating the biological aspect. So what I'm trying to show here is like highlighting that, of course, all of the trials have inclusion-exclusion criteria. And the basis of our design is to answer that question, that scientific question. And we do bring in create these inclusion-exclusion criteria to answer that question. However, there is obviously the influence of other real-world factors. So I just gave some examples. Obviously, inclusion-exclusion criteria are way more complex than what you're seeing on my screen. But like we use age, we try to recruit in different countries. Health authorities now require certain racial inclusivity as well. There's always something about the disease characteristics that you need to recruit. And then there's some exclusion criteria, which is on the right side here, around comorbidities, treatments that you can use at the same time or that you've used before. And all of these factors together, you could say they're kind of a surrogate, not exactly, of these types of real world factors. Like, the geographic inclusivity, the sites that we use in our trials are supposed to reflect the different cultural, sociocultural norms across the world. They're supposed to also factor in the different country-specific clinical care aspects that you have in different countries. So, and then healthcare access is a complex one. But the idea is that when you have all these other, the prior treatments, the concomitant medications, they to some extent do reflect the diversity of healthcare access that you can have in different countries. In particular, like some of the trials that I am currently running are in multiple sclerosis. And for those, what you see is that in a lot of countries in Eastern Europe and Russia and Ukraine, they haven't used a lot of different MS medications. But when you look at the American participants that we get, they have used an array of meds because of the differences in access there. So just to give you a slight idea of how these inclusion-exclusion criteria sometimes touch on these real-world factors, and to dive a little bit more into that, like, sure, we do this, like, what is the purpose behind it? I mean, our goal is basically to make these results, the eventual results that you get from the trial, make it generalizable to the global population, so to speak, but at least to the population that eventually gain access to the drug. Now, I touched on the various aspects of participant diversity, and to reiterate some of those in the context of how those translate into factors that impact the science of the drug. So I mentioned that we have geographic diversity and that like location, socioeconomic status. So like that is like the sites that we're bringing into our trial. We also obviously try to recruit across the spectrum of gender and sex. Now, the idea behind that, behind both of these nuances is like you want all of these countries and all of these people in to ensure that there is a diverse pool of genetics, because some of the genetic information is not even understood. And these different countries, when you bring people in and you try to ensure racial diversity as well, that you want to bring in folks who have different aspects of the environment, the food they eat, the level of activity that they have, their day-to-day, where they live. There are environmental influences that we don't understand even sometimes. So we need to diversify that pool to at least bring in those patients to indirectly measure that. And then, of course, for disease characteristics, we always define age criteria and certain types of disability levels, especially that's relevant in MS. So that's why it's listed here. So we try to target the entire spectrum of the disease as best as we can, not always, but these are the different factors that we use to make our results a little bit more generalizable by improving the biological diversity and indirectly touching on these real world factors. So all of this to say that I already told you my bias is that randomized controlled trials are a gold standard. I believe that my bias is rooted in reality, and that is because these trials are supposed to. Obviously, they're touched by global regulations, by the financial situation of the participants, the personal nuances of each participant. But these trials are not supposed to address these issues by introducing healthcare access solutions. But what they're supposed to do is somehow incorporate this by improving geographic diversity. They're not supposed to advocate for economic equality, but they are supposed to bring in participants of a diverse background to allow for some understanding of the impact. And then finally, when we say personalized medicine for us, that is more a genetic and disease biology thing. We can't factor in each country's like individual norms. So for us, it's more the genetics and the disease biology that we really want to incorporate within our design solutions. And so I don't actually think that these are limitations. That is the reality of the situation that we're in. And we are here to solve a certain problem. And then factor in these other elements so that at least the trial results are representative of the global landscape to the best that we can. And with that, I will hand it off to Rashad, who's going to disagree with me on all of these things. So, and I think the discussion will be after. So, Rashad, I can continue to drive.
Oh, you're going to continue to show the slides? Wonderful.
Yeah, I can do that, yes.
Okay, super. Let me just get myself ready with that. Okay. So first of all, Salat, thank you very much. This was wonderful of you, and you actually set the stage. So, Madam Chairperson, honorable guests here, the motion that we are debating is a very specific one. The motion is, this House believes that randomized controlled trials are the gold standard for research and learning. And here's where I pose a question to all of you. My question is, really, are they the gold standard for research and learning? And I'll answer it for you as well. I would say that it is the answer to the question of, are they the gold standard, is that it depends. What does it depend on? It depends on what we're trying to study and to learn about. Let me decipher a little bit of what my esteemed colleague Sana explained about randomized controlled trials. Randomized controlled trials are only one type of scientific experiment that's used in clinical research, in clinical trials. And the purpose of these trials are to evaluate the efficacy of a medical intervention. This is to say, does a drug work or not? That's essential. Does a treatment work? Does a procedure work, etc.? Now, given that that's the purpose, they have very specific properties or characteristics. The first one is that the hypothesis that we go into an RCT with is a very fixed hypothesis. It is, something that we cannot change as we work with it. The outcome is predefined, and the outcome is measured and, as Sana explained, statistically analyzed. There's a key thing here, which is the idea of randomization. That is, you've got one group, as Sana said, the intervention group that receives that intervention, and the control group, which does not receive the intervention, but receives something else. That can be a placebo, that can be the standard treatment that is currently being used, etc., which allows us to determine whether the effect that we are looking for can be attributed to what we're doing. Essentially, the whole idea of an RCT is to eliminate any bias or confounding factors by assigning different groups to different places and to eliminate all the other factors that could influence the result in order to make in order to decide on whether it works or not and the extent to which it works. Now, as such, the goal of an RCT is to provide evidence regarding whether a particular intervention works or does not work and the extent, the degree to which it works. So, is it a gold standard or not? Well, it's a gold standard for one type of research, not every type of research. It's only the gold standard if we're actually trying to determine whether a particular intervention or drug or diagnostic or drug causes that effect or not, and the degree to which it causes it. Now, let me step out of that space for a minute and ask the question, but is this all that we want to learn about? Is this all the research that we want to conduct? Or is there other research, other learning? Well, the other questions that come to my mind are things like, okay, if it worked, why did it work? If it worked, how did it work? What was the mechanism by which it worked? If it works, what is the context in which it worked? Or if it didn't work, what was it about the context that did not make it work? The other questions that also come to mind are, for different contexts, what factors make it work or not work? Why and how does that work? Now, for me personally, I'm an improver. I work on making healthcare better. And the question is, if I know that something actually works, if I know that I have an intervention that is efficacious, it has been established through an RCT to be efficacious, how can I make sure that everybody who needs it gets it in terms of the supply, the access, the adoption, the proper usage, in such a way that we can actually get the outcomes of interest? What I'm trying to differentiate between are two areas of knowledge and learning. One area that I call discovery science, that is, does something work or doesn't it work and to what extent? And the other area, which is the area that I work in, which is what I would call delivery science. That is, once I know that something works, how can I make it work for everybody who needs it in such a way that it will have the beneficial effect for the whole population that needs it? Now, that begs the question, surely if we've established that something works, then it must get adopted. Well, and this is where I really want to talk about. When I started in this arena, I remember an agency in here close by where I live called the Agency for Healthcare Policy and Research, which has today become AHRQ. All of you are probably familiar with the Agency for Healthcare Research and Quality. And I remember citing them so many times on a paper that they had produced where they said it takes about 17 years to transform 14% of evidence-based interventions that have been shown to be efficacious into everyday practice for the patients. In recent years, I've also looked this up to see if that has changed much. And I actually have not been moving the slides. Let me reference a couple of really important articles in this arena that I'm showing them on the slide right now. Sure, they're on the slide right now. But these are articles that basically tell us that it still takes about 17 years, which is the time lag, in translating what we know to work into everyday practice. And a more recent article in 2023 in JAMA also showed the very same effect. Now, you may ask, why is that? Why is it that when something has been established to work, it doesn't actually get implemented? Well, it's for all sorts of reasons. Some of them are human nature, but mostly are the way in which our systems are designed. And this is a reality. So there is a need to learn about how do we make something effectively work in a real life context. And this is called effectiveness research. It's a different type of research, which takes what's been established through an RCT to be efficacious, and then works with all the messiness of real life in order to make it effectively happen in the real world for the people who need it. This type of research is also called implementation science research, improvement science research, delivery science research, health services research, operations research. In fact, a colleague of mine once did a study and found over 200 different names that are used to describe this type of knowledge. This type of knowledge builds on not just quantitative methods like RCTs do, but also qualitative methods that try to get a deeper understanding of the whys and the hows. Also, it utilizes a lot of mixed methods research. In these instances, not only is randomization not required, it actually is not desirable and will interfere in the way that we get the results. In this type of science, what we try to do is to actually start with the places that are most likely to succeed, as opposed to the randomization, where there's prior history, where there's the right leadership, other factors that contribute positively, and develop it there, make sure that it can happen there, and then go in increasing orders of complexity, step by step, taking on more and more difficult ones and making it happen in those places. We're not out to prove that it works. We know that it works. We're now trying to make it successfully happen in a wide range of different places with different sets of circumstances. And interestingly, we find that what makes it work in one place doesn't necessarily make it work in the next place, and vice versa. We don't go in with a fixed hypothesis. In fact, our interventions, the hypothesis as such, are flexible, and we can adjust them, and we have to adjust them if we want them to work. So we start with any intervention that we think is likely to work, and we any way of implementing what has been established to be efficacious. So last year, for example, at this meeting, I showed how when we were working on antiretroviral therapy, half of the patients that needed it were not getting it. And so we tried different ways in which to get the patients to know their status, to enroll in care, and then to stay on treatment to do that. And to do that, we were testing different changes. Some changes worked for some patients, some changes worked for other patients, some changes worked for certain types of facilities, not others, and across countries. But over time, we were able to test and implement all sorts of different changes. We use what's called the Plan, Do, Study, Act cycle, the Shewhart method for learning and improvement. In these circumstances, not only don't we want to have fixed hypotheses, we want to be able to, if it works, and it works as well as we want to implement it. If it doesn't work, we may decide that we want to adapt it in some ways to make it work, or discard it, and so on, and continue to test different changes until we get the right types of results. Sometimes we can't use control sites as such in these types of experiments, but sometimes we do use comparisons. And some of our comparisons are actually, because we know something works and we want to take it to scale, we may not be able to go to scale right at the beginning. So we use our initial sites as the testing ground, but measuring the data in sites where we're not intervening. And then as we go to intervene, then we compare the results, what's called a stepped wedge design type of setup, which enhances our ability to know that all these different things that we are putting together have made it work. So let me summarize. In implementation science or improvement science research, what we're trying to do is to take something that's already been established through an RCT to be efficacious and to make it work in real life by, first of all, testing it and adapting it, and also discarding it if it doesn't work. There are some commonalities. One commonality is that the outcome is predetermined. We know that our objective is to make something happen effectively in the real world, and that doesn't change. So that's sort of in common with RCTs. There may be some similarity. Sometimes we do use comparison group, but they're not actual strict controls like you do have in an RCT. What is very different? What's different is that the hypotheses are not fixed. We use the Plan, Do, Study, Act cycle to test different changes. And some changes we may know about ahead of time, and others we develop as we go along and as we learn. We don't randomize. We actually start with the easiest and go to increasing levels of complexity. And then the analyses are done in real time. That is, we collect the data literally on an everyday basis. As we're testing, we look at the results and we decide based on that. For this type of research, not only are RCTs not the gold standard, they're actually inferior to the other types of research that are needed for this type of work. With this, if I may hand over back to our esteemed chairperson, please, Suzanne, back to you.
Thank you, Rashad. And what great arguments, both sides. And I can relate very much, Sana, because as you mentioned, I worked on ultra-rare diseases, and the complexity is on another level when you're running a study with only 7 patients. So imagine the endpoints, every data is very crucial. And actually, that's where also things like diversity, and now we have new regulations, and actually the regulators are demanding that we run clinical trials diverse. But then you do have the consideration for geographical areas. So what do we do about China? China has been classified as a non-diverse country. So what if the patients are there? So there's so many considerations when it comes to the clinical trial design. and the data. And also, I can really very much relate to you, Rashad, because in quality, we always use the plan, do, check, and repeat cycle. And that's, the foundation. So is it a gold standards? Are RCT the gold standards? And maybe not for everything. So I can see both sides, but I want to open now the floor to all of you guys on the call. What is your thoughts? What's your comments? Do you have any questions?
Yeah, I can just maybe get the group going. What really I found particularly striking is Rashad's comment on the speed, on the speed of the RCTs and how many years behind it may leave some patients, without the opportunity for any of these treatments as well. So that was something that I've also thought about quite a lot as well. A great debate and a lot of great points both sides. Yeah, Suzanne, back to you. But that's my comment. But please.
No, I agree. The speed, the investment, and after all, it might fail. So it's
Right.
it's a lot of considerations. And I was discussing with Rashad, you can also do the wrong thing in the right way. So it's very interesting, the dynamics of where the future is going to take us and how we run our clinical trials. Any other thoughts or comments?
I have
Yes,
thought of
please.
the
Yeah.
Yep. So the speed, yes, what the heck, 14 years or 17 years, people even everything changes by the time it hit the ground. So I'm hoping that health avatar and AI can do simulation in a way that it simulates the real world and the virtual world, which I think Meta, Facebook are working on something similar to that. I hope
Yeah.
that will fix this huge worldwide problem. Are you seeing any of these applications, Rashad and I think Sana?
So, and maybe I can start because I think in the context of designing clinical trials, for instance, I can start there, and then maybe Rashad can speak to more of the real world implementation side of it. So there are elements of AI that we're using, but I just presented a very basic design, right? And what is being done now is like using these technological aspects and creating, for instance, like a virtual control arm. It's still early phases, but it's something that is being looked into. So there's a lot of adaptations that are being incorporated, and I get it. Like the rolling out of the drug after the trial ends is this different type of gap period that Rashad alluded to, but there's also the duration of the trial itself. MS trials, for instance, can run up to, like my trials, for instance, run up to 3 to 5 years. That's the duration that we have. So the trial duration itself is something that we work on optimizing, and it varies. For some other drugs, it's smaller than that. But to answer your question, more or less is that we're using elements of AI in different ways, not exactly the ChatGPT version of it, but certain aspects of it to create virtual control arms, for instance, to help us. And we do that using real world databases. So we are trying to connect those two so that if it's a rare disease and you want everybody to get the drug, you just compare it to like this virtual database. There is like some work to be done to win over the health authorities to this approach, but it is definitely doable. Like Bayesian methods, for instance, that's another statistical method that we use to kind of offset this. But Rashad, I don't know if you want to add anything.
So we're in the early stages of using AI, and already, I mean, we're using it in terms of just being able to understand what the reality is. The amount of data that we can gather is much more than what it used to be prior to the advent of AI. I mean, if you were using Google searches and literature searches, it took forever. Now it's much more straightforward. So that's one place where we are definitely already seeing the benefit. I think going forward, we'll also have the benefit of being able to use it to share knowledge on what works, what doesn't work in different places. Now, when I talk about that, it sounds like, okay, isn't that knowledge already there? There's different types of knowledge, and the knowledge I'm particularly interested in is what's called tacit knowledge. So there's the explicit knowledge, everything that you read about that's in papers. That's, you know, when you write your article, you're going to spell out the things that. Now, if I really want to be able to do what you did, I have to dig deep, and getting, you know, you have to ask the question multiple times in order to get down to that knowledge that is very profound in nature about how you were able to achieve that. Now, in managing knowledge, we've honed in on how do we create the conversations that generate that type of tacit knowledge, and then how do we harvest the tacit knowledge in a way that makes it usable for others. There's a nuance here, which is what worked in a particular place in a particular instance may or may not work the same way somewhere else, but the change concept behind it is usually applicable. So one of my best stories about change concept, if I may take a couple of, just another minute to explain.
Not so many minutes, Rashad. One
Okay,
minute.
1 minute.
Two
Now,
more.
these were the early days of nevirapine in Rwanda, and I remember trying to get the spouses tested for HIV/AIDS, the spouses of women who were pregnant, about to deliver, in order to. So we were testing them for HIV, but we also needed to test the spouses, and we whether to know whether we administered nevirapine at the time or not. And I'll never forget that. You know, only about 2 out of 10 spouses were showing up to be tested. And then one midwife was able to show us how 80%, like 8 out of 10 were coming. And we said, how did you do that? We're in a big meeting. It's called Collaborative Improvement. And she started to explain. She said, well, I talked to the woman, and she said, if the doctor can write a note to my husband, I'll, you know, because she told him and he wasn't listening. She took that note, gave it to her husband, and the husband showed up. And then the nurse started to test it with other women, and it worked. I'm standing there listening to this, and I remember looking at my regional director for Francophone Africa. And I said, there's something really, I don't understand this. Why would he go based on a note from a doctor he hasn't even met, when his wife, but not listen to it? I said, oh, this is a cultural thing. In this part of the world, it's an insult to refuse an invitation from a man of superior, another male of superior status. And I'm like, oh my God, this is the change concept behind it. So it's not just a note, it's a text message that works. It's also the phone call that works. And we started to test all of those, and all of them work. But that's the type of tacit knowledge you need to get out, because once you have that, the world is your oyster. You can start playing with that concept in all sorts of ways and getting the result that you want. Now, could we have AI help us disseminate that? Absolutely. And that will be something coming. I'll stop here. I can go on forever with my stories.
Thank you. Thank you, Rashad and Sana. And I'm just going to go to the left of my screen to Jolie. Yoli?
Yes, thank you. So I just have a comment. So I agree with Rashad. I think that, you know, the clinical aspect, you know, I mean, all the new studies really show that, you know, they're just half the story, right? Or even less than that. A lot has to do with, you know, all of the other social determinants of health, and especially, you know, there's regional aspects in terms of traditional medicines and how they live and how they treat their diseases, right? So I think that unless the full view or the full person view is not taken into consideration, we will not find solutions that are, I would say, adoptable in mass, right? So if you look at the trying to get the economic benefit, if we continue to fund research and clinical trials that may only, you know, be accessible by a handful and work only for a few, we're not really addressing the broader issue. And lifting everyone. It's my
Thank you
thought.
for that. Yeah, true. And Mark?
Hi, can you hear me? I had to switch to my phone. Can you hear me?
Yes, we can hear you.
Okay. Yeah. So to answer the question, are we speaking about randomized controlled trials in the classical sense of the multi-year, the strict sense of an RCT? Because so to me, there's no gold standard in clinical evidence generation. It's very much tailored to what you're trying to achieve and the existing evidence, and so it becomes a rabbit hole. It becomes a great debate topic, because when you try to say there's a gold standard when one potentially doesn't exist, of course we'll be debating all day. But so to answer the question, are we talking about a classical randomized control trial structure, or are we thinking about the evolution and creating randomized controlled trials from synthetic data, from virtual patients? How innovative are we being here with this debate?
It's everything, Mark. I think you are posing a question here. So it is considered, I just gave examples of the classical design, but the idea is that clinical trials as they stand, usually they are controlled in some sense, whether it be through a Bayesian method, through a virtual arm, what have you. It can be adaptive. I've done a lot of adaptive design myself. So it's the overall question is everything that randomized clinical trials represent. Are they sufficient, even with their various hybrid methodologies and incorporation of technology? Are they still sufficient to address the issues that Rashad alluded to? That's the idea behind it.
Yeah, I think there's a bit of a gray area here, but I think from a scientific perspective, the randomized control trial approach, the scientific and logical rigor, the statistical methodologies to actually answer the question, for me, I would say that fundamental approach scientifically would have to be the gold standard. However, the old school 7-year, clinical, multi-center clinical trial is too slow. So we need to be more innovative with how we approach RCTs. And I'm really interested in virtual care and like synthetic data, and how can we, if you have 7 patients, can AI generate 70 more for you? That sort of thing.
Yeah. It's an area of interest of mine too, Mark. So yeah, we're watching now the space, and there's a lot to come, because in 50 years, everything we are doing, even all of this, are we going to have the standard protocol the way we have it with all these protocol amendments? And it's not going to be that way. And I'm very curious how it's going to map out. And I just want to give a warm welcome, because I just see the comment from Tim that we have Chris from the patient advocates group. So thank you for joining us. And I'm really curious to see all of your votes at the end, but we're just going to give 2 minutes for both Sana and Rashad to summarize before we go into the voting. So to you, Sana.
Okay, thank you, Suzanne. And thank you for this really interesting conversation, and for everyone that pitched in with their comments. So I hear the comments, and I hear like Rashad's side of the story, and I will say that it's not that necessarily we disagree. We see the problems on both sides of this. However, my stance still remains that clinical trials, with all of their complexities, their innovations, their different adaptive hybrid designs, do represent the gold standard to answer the scientific question. And I still know that those other issues do exist, and they are still a problem that needs solutions. However, I think that is a problem not to sound crude, but it's somebody else's problem to solve. And it's people like Rashad that need to come in and optimize that and take our data and run with it. I agree, 17 years is too long. But guess what? That 17 years might be in certain countries only. Those countries, and it might be that there is a realm of the way that the system of getting drugs approved that needs to be changed and better regulated to allow for greater, more expedient implementation. However, I think that the scientific method, which is well controlled, rooted in science, rooted in statistics, remains the gold standard to understand the biology, and all those other factors exist. And that is why we have created adjustments to our recruitment tactics and whatnot to improve the diversity. And we need to continue to improve that. And we need to continue to use data that is real or not real, but still rooted in reality to optimize that. But I think that the randomized control trials remain the standard. And with that, I hand it to Rashad.
Thank you, Sana. So let me say something. I'm a physician. I'm a public health specialist. I have worked most of my life in the area of improving healthcare. And over the years, I have seen the power of taking something that is known to work, that has been established through an RCT to be efficacious, and then to make it happen in real life for every patient, every time it's needed, in a reliable enough way that produces the outcomes that we are all seeking to get. I've experienced it over and over. I've experienced it in over 100 countries in pretty much all of the health areas that we can think of. Now, I've often been asked the question, well, where's the RCT to prove it? And literally that got me on a mission, because intuitively, I knew that RCTs cannot work for this type of learning and for this type of research. And it took a several-year journey that culminated in convening a Salzburg Global Seminar, putting 60 people together, basically researchers, improvers, policymakers, people from all walks of life to come together, locked up for a whole week together, and with a design that took a couple of years to actually prepare for, and to answer the question, what are the research methods that are appropriate for what type of research? And as a result of that, we published a supplement of the ISQA journal, the International Society for Quality in Healthcare, specifically addressing this topic area. As a result of that, I became much more acutely aware of the fact that RCTs are definitely the gold standard for clinical trials. That is, let me explain it simply. I would not take a tablet that has not been established to be efficacious through a randomized controlled trial. But the fact that it's been established to be efficacious doesn't mean that everybody who needs it gets it. There is a whole lot of other research, a whole lot of other learning that does not lend itself to the RCTs. And in fact, whenever they've tried to conduct an RCT on that type of research and learning, it has actually failed. A classic example is a Lancet article that basically showed that dexamethasone does not work in premature babies. Well, we all know it works, and we know that that study basically used the right technique to answer the wrong question. And that's what I'm referring to. So when somebody comes to effectiveness research and says, where's the RCT on effective research? I say, not all research lends itself to RCTs. And the motion that I am opposing specifically is not, are they the gold standard for clinical trials? Are they the gold standard for research and learning? They are the gold standard for one type of research and learning, they're not the gold standard for other types of research and learning. In fact, they are inferior as a research methodology to other types of research and learning. And this is the motion that we have at hand. I will stop here, and I look forward to your vote.
Thank you, Rashad. Please, I would ask you all just to go to your application, whether it's on your phone or on your PC, and cast your votes.
Yes, and I'll show that actually in a second here as well. So if you're on the mobile phone or on the laptop, just make sure to scroll down on the left where it says resources and look for the poll button. Just one moment, I'll share it in this moment. Here we go. So if you're on the web app, which I recommend highly, go to the resources and click in the polls here. I noticed that there are some of you already that have voted. So let's
Where are
see.
the resources? It's not visible.
Here on the left. So polls. The actual you're looking for polls on the left side.
Are you sharing your screen, Tim?
Yes. Oh,
Okay.
you're not seeing
We
it?
didn't see it. No.
Oh, okay. Okay. Okay, so let's see. So there's your motion. So here we go. So there are 3 responses. So we're going to wait for some more. And then we're going to view the results here. Okay, so the votes are coming in, and we're kicking off the competition already. So thanks for those of you who are voting. We heard quite a lot comments as well. Oh, 7 responses. Okay, Suzanne, let me know when you'd like me to display the results. I believe you should be able
Just
to.
give it.
You should be able to
Yeah,
do
I
it.
can see it. I can see it. Yeah.
Oh, you can see the results as well. So please feel
Yeah.
free to share it as well.
I'm waiting for more to vote because I can see
okay.
more participants. Yeah. Just give us 5 seconds more. All right. So the winner is, drum roll, Sana for the motion. You win, but it's a very, very close because we got just 1 vote over against the motion.
Wow.
And I have to say, though we are voting for a winner, I want to say both debaters gave really compelling arguments and very interesting takeaways. So thank you both, and thank you everyone for very engaging and, feedback and contribution. I very much enjoyed this debate too. And I wish you an amazing rest of the day and enjoy the Olympics. Thank you.
Thank you. Thank you, Suzanne. Thank you, Suzanne, and everyone. Rashad, Sana.